Hello, and welcome to this in-depth exploration of the history of bioinformatics and genomics. This field, where biology and computer science converge, has transformed how we understand the building blocks of life. In this video, we'll traverse through the historical milestones, from the early days of genetics to the sophisticated genomic analyses we conduct today. Buckle up for a journey through time, exploring the breakthroughs that have shaped our current understanding of biology. The foundations of genetics. Introduction to Mendel's work. Gregor Mendel, often considered the father of modern genetics, was an Austrian monk who conducted pioneering research in the mid-19th century. Mendel's choice of pea plants for his experiments was not arbitrary. Pea plants have distinct, easily observable traits such as flower color, seed shape, and pod shape. Mendel meticulously crossbred pea plants and observed the inheritance patterns of these traits over several generations. Mendel's experiments. Mendel's experiments were methodical and innovative. He started by focusing on a single trait at a time, such as seed shape, round versus wrinkled. By crossing plants with different traits, Mendel was able to deduce that traits are inherited according to specific ratios. For instance, in his experiments with pea plant seed shape, he found that when he crossed plants with round seeds with plants with wrinkled seeds, the offspring, F1 generation, all had round seeds. However, when these F1 plants were crossed among themselves, the wrinkled trait reappeared in the F2 generation, following a 3-to-1 ratio of round to wrinkled seeds. Principles of inheritance. From these experiments, Mendel formulated two key principles: the law of segregation and the law of independent assortment. The law of segregation states that each individual has two alleles for each trait, one inherited from each parent, and these alleles separate during gamete formation. The law of independent assortment states that the inheritance of one trait is independent of the inheritance of another trait. Mendel's impact and legacy. Despite the groundbreaking nature of his work, Mendel's findings were largely ignored during his lifetime. It wasn't until the early 20th century that his principles were rediscovered by other scientists such as Hugo de Vries, Carl Correns, and Erich von Chemack. Mendel's work laid the foundation for the study of genetics, providing a framework for understanding how traits are passed from one generation to the next. Mendel's work was revolutionary, providing the foundation for modern genetics. His principles continue to influence biological research and applications today, demonstrating the lasting significance of his contributions to science. The discovery of DNA and cracking the genetic code. The discovery of DNA structure. The discovery of the DNA double helix was a landmark event in the history of biology. In 1953, James Watson and Francis Crick, with crucial contributions from Rosalind Franklin and Maurice Wilkins, elucidated the structure of DNA. Using X-ray diffraction images produced by Franklin, Watson and Crick proposed that DNA is composed of two strands forming a double helix, with bases paired in a specific manner: adenine with thymine and cytosine with guanine. X-ray diffraction and Franklin's contributions. Rosalind Franklin's X-ray diffraction. Images of DNA, particularly photograph 51, provided critical insights into the helical structure of DNA. Franklin's meticulous work, including her application of X-ray diffraction techniques, allowed her to capture high-resolution images that revealed the helical nature of DNA. Although she did not receive the recognition she deserved during her lifetime, her contributions were crucial to the discovery. The significance of the double helix model. The double helix model explained how genetic information is encoded and replicated. The complementary base pairing mechanism ensured that DNA could be accurately copied during cell division, a fundamental process for heredity. The structure of DNA also suggested a mechanism for genetic mutation and recombination, key processes in evolution and genetic diversity. Deciphering the genetic code. Following the discovery of DNA's structure, researchers turned their attention to understanding how genetic information is translated into proteins. The genetic code consists of sequences of nucleotide bases, triplets, that specify amino acids, the building blocks of proteins. In the 1960s, scientists such as Marshall Nirenberg and Hargobind Khorana deciphered the genetic code, revealing how sequences of nucleotides correspond to specific amino acids. The impact on molecular biology. The discovery of DNA's structure and the genetic code revolutionized molecular biology. It enabled scientists to understand the mechanisms of gene expression, regulation, and mutation. Techniques such as recombinant DNA technology and polymerase chain reaction, PCR, emerged from this understanding, facilitating advances in genetic engineering, cloning, and genomics. The first genome projects and the Human Genome Project, early genome projects. The concept of sequencing entire genomes was first explored in the late 20th century with several pioneering genome projects. These projects aimed to map the genetic sequences of various organisms to understand their genome's structure and function. One of the earliest genome sequencing efforts was the sequencing of the bacterium Haemophilus influenzae in 1995, which was the first free-living organism to have its entire genome sequenced. The Human Genome Project, HGP. The Human Genome Project, initiated in 1990, was one of the most ambitious scientific endeavours in history. The goal was to sequence and map the entire human genome, which consists of about 3 billion base pairs of DNA. The project involved an international collaboration of scientists, researchers, and institutions, utilising cutting-edge technologies and computational tools. Technological advances and challenges. The HGP required significant technological advancements to handle the vast amount of data generated. Techniques such as shotgun sequencing, where DNA is randomly fragmented and then reassembled using computational methods, were developed to speed up the sequencing process. The project also faced numerous challenges, including data management, computational analysis, and the integration of diverse data sources. Data analysis and interpretation bioinformatics played a crucial role in analysing and interpreting the data generated by the HGP. Researchers developed algorithms and software to assemble the fragmented DNA sequences into a coherent genome map. The analysis also involved identifying and annotating genes, regulatory elements, and other functional regions within the genome. Impact on medicine and research. The completion of the HGP in 2003 had a profound impact on medicine and research. It provided a comprehensive reference genome for human genetics, enabling researchers to identify genetic variations associated with diseases and traits. The project also paved the way for personalized medicine, where genetic information is used to tailor treatments to individual patients. The rise of bioinformatics and modern genomics. The evolution of bioinformatics. Following the completion of the Human Genome Project, bioinformatics emerged as a critical discipline for analysing and interpreting the vast amounts of genomic data generated. Bioinformatics combines principles from computer science, statistics, and biology to develop tools and techniques for managing and analysing large-scale biological data. This field has become essential in understanding complex biological systems and has led to significant advancements in various areas of genomics. Bioinformatics, tools and techniques. Bioinformatics encompasses a wide range of tools and techniques designed to analyze genetic information. Key tools include sequence alignment programs, gene prediction algorithms, and protein structure modeling software. For example, sequence alignment. Tools like BLAST, Basic Local Alignment Search Tool, allow researchers to compare nucleotide or protein sequences against large databases to identify similarities and functional annotations. BLAST has become a fundamental tool for identifying homologous sequences and understanding evolutionary relationships. Gene prediction. Algorithms such as GeneMark and Augustus use computational methods to predict the locations and structures of genes within a genome. These tools analyze DNA sequences to identify coding regions, introns, exons, and regulatory elements. Protein structure modeling. Programs like PyMol and Chimera enable researchers to visualize and model protein structures based on experimental data and computational predictions. Understanding protein structure is crucial for elucidating protein function and designing targeted drugs. Applications in genomics bioinformatics has revolutionized genomics by enabling the analysis of complex datasets and providing insights into genetic variation, gene expression, and functional genomics. Some notable applications include genomic variation analysis. Bioinformatics tools are used to identify genetic variants, such as single nucleotide polymorphisms, SNPs, and insertions, deletions, indels, which are associated with diseases and traits. The analysis of genomic variation helps in understanding the genetic basis of diseases and developing personalized medicine approaches. Gene expression analysis. Techniques such as RNA sequencing, RNA-seq, generate data on gene expression levels across different conditions or time points. Bioinformatics methods are used to analyze these datasets, identify differentially expressed genes, and gain insights into gene regulation and cellular processes. Functional genomics. Bioinformatics integrates data from various sources to predict the functions of genes and their interactions within biological pathways. Tools like KEGG, Kyoto Encyclopedia of Genes and Genomes, and Reactome provide comprehensive databases of metabolic and signaling pathways, helping researchers understand the functional roles of genes and proteins. The rise of bioinformatics has transformed the field of genomics, providing essential tools and techniques for analyzing complex biological data. As the field continues to evolve, bioinformatics will play a central role in advancing our understanding of genetics and improving medical research and treatment. In this journey through the history of bioinformatics and genomics, we've explored the foundational discoveries that paved the way for modern genetic research. From Mendel's early experiments to the groundbreaking Human Genome Project and the rise of bioinformatics, we've seen how these milestones have transformed our understanding of genetics. As we look to the future, bioinformatics will continue to play a pivotal role in unraveling the complexities of life and advancing medical research. Thank you for joining me on this historical exploration. In our next video, we'll delve into how bioinformatics is applied today to address contemporary challenges in science and medicine. Stay tuned and see you next time.